My name is Markus Kolbe. I will provide the DECODON Academy webinar today about experimental design and statistics for 2DE data. Let's first begin with more global aspects. What is the aim of your project? What are the project goals? Of course, you want to answer a biological question that is of interest for you, and then you want to, of course, create a publication or a poster with creating or preparing results ready for publication. You have other features in Delta 2D, such as color coding or exporting images as PowerPoint slides. That should be rather easy, but is part of a different webinar. Finish within scheduled timetable. This is, of course, very important since some deadlines approach, project deadlines maybe, and of course always budget is limited, so you should finish your project and your analysis within a certain budget. This leads us to other aspects, and that means you should work efficiently and you should check yourself whether you are, for example, able to produce good gels repeatedly. Is the quality of the gels varying too much? Is the technical variance small enough so that you really can rely on the results? That technical variance is smaller than the expected biological variance? Did you define good milestones? And furthermore, if they are not met, are you prepared to stop your project? Sometimes it might be better to stop a project and disband it and stop investing more budgets and time also into a project rather than starting again, learning from others, maybe using their experience and getting started again with the project with different maybe consumables, other protocols or whatever. Another aspect is, will I keep records? Since whatever you learn, whether the project is successful or not, doesn't matter. You should record your results and what you have learned, since if you are asked from colleagues, from others working on the same in the same area, you should be able to just let them know what you have learned and where you had problems or not, so that others do not run into the same difficulties and can make use of your good experiences. It's always good for your image if you can answer questions and show your experience so that you become more and more a multiplicator for the technology. Then coming to the more operational step of project planning, you should always very well design your experiment, and before doing so you should well focus your biological questions. It doesn't make sense to just say, I want to learn something about my organism of interest. You should focus your question and then design the experiment with respect to that. Then you should also randomize when taking samples. You should also try to anticipate problems that might arise during your experiment. Buffer is maybe finished during the project, so that you should have enough at hand. Maybe if you run multiplex experiments using an internal standard, the standard mixture might not be sufficient and run out during your project. So you should be aware of possible problems that might arise, and some will arise during your project. You should use more replicates instead of additional factors, since with limited budgets you should spend your funds, your budget and your capacity into reliable results rather than complex results. Complex results are not so easy to handle, they are difficult to interpret, and if they are not reliable from a statistical point of view, then it's difficult to publish them. So it's much better to get reliable, significant results with a smaller focus. You should also prefer biological replicates against technical replicates, since maybe using a pilot experiment, which is the last issue here, you could find out your technical variance, and if it's small enough, you maybe can really avoid technical replicates and concentrate on biological ones. Yeah, and the technical variance should be more or less constant over time if you use protocols and standardized workflows always the same time, and maybe also consumables from the same batches. Then the experiment itself. In our example here we have cell lines and two factors, or the cell line, one cell line which is split into 4 sublines. One factor is the cell line splitting, and the two factors, the second factor is time. So we will get a control and 3 different treatments for 3 different subcell lines and 3 points in times. That means we take samples at the beginning at 0 minutes, after 30 and after 60 minutes. And in this experiment 4 replicates have been made for each sample, and in this case these are technical replicates. And if I say we, then that's not really correct. DECODON doesn't run an own lab. We got all the data from Christian Scharf. He's in Greifswald at the university and has helped us with this data set, which is large enough for advanced statistical analysis. Running 48 samples means if we use a multiplex approach using an internal standard, we need 24 dyed gels, and of course we then have to think about how to assign samples to the gels and to the dyes that we use. Just as an example, this is only the table for the wild type. For the first original cell line we have 3 points in times with 4 replicates each, and they are all spread across the first 12 gels using dye swapping, meaning every second sample is labeled with G200 and with G300 respectively. That means that we do not have an effect of the dye selection for a certain sample. Of course we get finally then 12 images for these 12 samples. For the other cell lines this is similar. We just have to add the remaining samples to the gels, to gel 1 to 12. We add in the opposite way the remaining open dyes to certain samples so that we fill the 24 gels times 2 dyes, and then also we have to have enough standard sample, that means an average mixture of our 4 cell lines across all the points in time. This needs to be sufficient in amount for all the 24 gels, since we have to add the standard to all these labeled with the G100 dye, or Cy2, Cy3, Cy5 using GE dyes. In total we will get 48 images for the 48 samples and plus 24 images for the standard images, for the standard samples. This is how the setup is looking like then. The two factors, one is over time, the other is over the cell lines, and each replicate group then is colored in a certain color. This is just a screenshot taken from Delta 2D. And if we want to now do a certain comparison, this is typically a comparison of 2 groups, and whichever 2 groups you select, you have changed at least one of the 2 factors that we are analyzing. Keep this in mind, since we will cover this during the t-test in a minute. The recommended workflow of Delta 2D is that we set up a project, we warp images, we detect and quantify spots, of course, and transfer the spots to get complete expression profiles. And then we come to the step which is covered today in this webinar. We analyze them to find interesting expression profiles and to be able to ignore the boring expression profiles that we do not want to publish. Finally, after that, You can, of course, present results in your publication, in your presentation, on a conference, in your talk, and the technologies that we offer for this are covered by a different webinar. This is how in Delta2D quantitative analysis looks at the beginning. You get data in two ways. One is the quantitation table that includes data across the complete experiment. That means for all the gel images and all the spots. And there you already have access to, for example, standard deviation within certain groups, the ratio between groups, the p-value for the t-test of a group pair. This is just a basic possibility to analyze your expression profiles. The more advanced is the statistical toolbox, where you have access to sophisticated statistical methods such as clustering methods, Pavlidis template matching, t-test ANOVA, two-way ANOVA, non-parametric tests, and also principal component analysis. Non-parametric tests become more and more important since they are the equivalent tests to t-test ANOVA and two-way ANOVA without having the need of normally distributed data. So the first thing is the quality control, and we can use principal component analysis for that. What is principal component analysis? Imagine you have a vast amount of data in your table and you want to get an overview which columns of the table, that means which gel images, are similar to each other. And you would expect that replicates, of course, are very similar to each other. And of course, making a decision whether it is similar or not, we could use every complete column and compare them. But that means for maybe 2000 or 1200 spots, you have a 1200 dimensional space, and for each column, for each gel or sample, 1 point in this multidimensional space. And then you should you have to make a decision how close these data points are in this multidimensional room. And this is not very handy. It's very difficult and it's not easy to find similarities there. Principal component analysis is reducing this complexity and is projecting this multidimensional space into a 2D or 3D space, providing the maximal possible variation between the data points. So this is a transformation of coordinates, and the resulting principal components are the resulting 3 or maybe 2 first principal components or coordinates that we use to spread the data as much as possible. So if it's true as here, we have a number of data points, and if they are well separated like here, so the colors stand for replicates, then we have done a good job, since all these nice clusters show that the similarity within the cluster is higher than across the clusters. Or in other words, the variation between clusters is higher than within clusters. Let's have a look at this in Delta2D. Here we go. This is this statistical toolbox. As I said, access to clustering methods, Pavlidis template matching, t-test, ANOVA, non-parametric tests, and principal component analysis. And that's what we want to use now for the first quality control. We want to cluster the samples. We could also cluster the genes, the expression profiles, by the way, but at the moment we want to do quality control to find out how similar the samples, the images are. I say, okay. I get a new result branch here in my main view in my tree, and get a 3D view as it is shown now. I hope that it is transferred properly to your screen. And we also have 2D views for principal component 1 against 2, 2 against 3, or 1 against 3. And having the 3D view means I can also rotate this view to really see how close are the different data points with the same colors to each other. And as we can see, there are shades of blue, shades of green and red, and it seems that within each color they are very close together, and also within shades of blue, for example, and also green. There is some differences for here, some red, orange and yellow ones, but I think you will agree that these data points are clustered well. Of course, there is some similarity between shades of blue, but the similarity within the clusters is very good as well. It would be difficult if we would see a blue point not within the blue cluster, but maybe together with the yellow or red ones here on the right hand side. Then we would see it and see an outlier, and we would have to review this image and have a look at it to make a choice. Should it be excluded, since maybe the first dimension did not separate the sample very well? Is there any problem with the electrophoresis? Has the imaging been problematic or whatever? So you can find out outliers, but you should then make your own decision what to do with it. After that, we are interested in certain changes between two groups, and that's of course up to your biological question, your matter of interest, what questions you have and what test you should use then. The simplest way is to use a t-test to compare two different groups. And comparing two groups means you have changed one factor, and the t-test says, okay, you can compare two groups that belong to the same factor, but have a variation within one factor, but not for the second one. So let's check that in delta 2D. I open the t-test dialog, and this is now providing me the groups as we have defined them in delta 2D, in the light table, for example. And here I can just make my decision which of these replicate groups should be my group 1 for the comparison. Maybe I'm interested for cell line that has been applied to treatment 1 and want to compare the point in time 30 minutes and 60 minutes to each other. Then I use 30 minutes as group 1 and 60 minutes as group 2. Then there is a number of parameters that we can adjust. For example, we can use Welch approximation, which is, well, I would say recommended, since we otherwise would have to check whether the variances within each group is equal or not. And then we can also decide for p-value parameters. We have to use p-values based on permutations. That's something more advanced, but that's a good choice, since it avoids problems coming with the data distribution. If we just have 4 replicates, then this definitely makes sense. The overall critical alpha, the critical p-value, could be 0.001 or 0.005. There are different ways to define that, and you can play with that. You can repeat the test with different settings, of course, and keep the results from the different t-tests that you have been used. So I can just try 0.001, and then maybe if there are just 2 or 3 interesting expression profiles, I can try with 0.005 again to find out whether I find a more interesting number of expression profiles. Then for discovery control, we recommend not to use Bonferroni, standard or adjusted, doesn't matter, because it's very strict and you would lose a lot of maybe interesting expression profiles. But we would like to use force discovery control. This is important to do this because we have here maybe 1,000 or 2,000 spots in our experiment, and that means we repeat the t-test for each single expression profile again and again. And as you are aware, the more often you play the lottery, the more often you win. But you win just by random, not because you have known the numbers in advance. And this is the same here. The more often we do a t-test within a certain experiment, then the more often we will find false positives just by random. This is just because of biological variance and distribution of the data of the expression of proteins. So we need to do that. Maybe I can just show this in my presentation again. The t-test has a certain approach. It is looking for the mean of expression levels into groups, and is the difference of this mean significant or not. The null hypothesis is saying the mean is the same, and if we can reject this, then we know the mean is not the same, with a certain level of significance or certainty. ANOVA, by the way, has a similar null hypothesis. The mean of expression levels in all groups are the same, and it is rejected by any change that can be observed between any pair of the groups that you are studying. And you have to, of course, follow this up and see where you have found this difference that leads to the rejection of this null hypothesis. Multiple testing problem means you really have a number of tests, and you have to avoid that you run into problems by identifying false positives. The larger the experiment, the more spots you have, the more t-tests you will run, and that means by random, the more false positives you will find. For example, if you use a critical p-value of 0.005, 5% so to say, and you have 1,000 expression profiles, you should, just by mathematics, by statistical analysis, find 50 expression profiles to be significant, but this is just random finding. There's no difference between the organisms, the cell lines, the points in time at all. It's just because the data is distributed over a certain data range. And this can be addressed by using force discovery control. Let's go back to delta to E. Here we go, and we can choose whether we want to have a maximum of 10 false positives in our findings, or a certain proportion of maybe 5% among our findings. I say, first we also choose to construct a hierarchical tree for the results. I say OK, use complete linkage. I don't want to use the sample tree clustering because I don't want to resort the columns. I say OK, and then we see there is a certain number of expression profiles, and if I just count them, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, it's exactly 10, and I expected a maximum of 10 false positives using the false discovery rate, and that means that all of these 10 could be false positives. This is just mathematics and doesn't say something about your biological background, but you can of course follow all these 10 up and say, okay, they might be false positives, but I cannot be sure. They match my biological theory, so I really do mass-spec for all these spots and follow them up to learn more about them. That's of course up to you. And then we get the same view as hierarchical tree, where you can see that, for example, here we have a certain typical behavior which is different to this one. You can see this by the colors. By the way, the columns here represent the different images coming from the samples. First we have the control samples, then treatment 1, treatment 2, and then also treatment 3. They are ordered by the time points, and within the time points we have 4 replicates each. And we have studied treatment 1, 30 minutes against 60 minutes, and we should see that there is a certain step variation between this block here and this block here, because these are the images for treatment 1 30 minutes, and these are the ones for treatment 1 60 minutes. So with this, you easily find your expression profiles of interest. You can run the test again, you will get a new branch for this new result, and then you can compare what you have found. Of course, if you click here inside, you will get the same selection of spots in the dual view and in the other views in Delta 2, in the table views, for example. Force discovery control is nice to do that. Avoid one-for-owning, what I have said, it's very strict. And then we can also compare this to a non-parametric test, which does not demand for the normal data distribution. So data doesn't necessarily be normally distributed. For this, we have 3 different tests here mentioned, which are alternatives to the t-test, one-way ANOVA or two-factor ANOVA. We use the Wilcoxon-Mann-Whitney test to be able to do the same as we did with the t-test. To replicate groups one factor, we vary just the factor time. Non-parametric tests can be here. I use the Wilcoxon-Mann-Whitney, press next. Here I could now define group names. I just leave this by default. And then we have to assign each image to group 1 or to group 2. By clicking here I can do this, or I exclude the image from my analysis. I have prepared this setting for you. I have a setting which assigns all the images to a certain group, to group 1 or group 2. I choose this one, it's the t-test, treatment 1, 30 minutes against treatment 1, 60 minutes. I open this one, and as you can see now, all images are excluded, but treatment 1, increase the size of the treatment 1, 30 minutes, is defined as group 1 for the Wilcoxon-Mann-Whitney test, and treatment 1, 60 minutes for group 2. All the others are excluded from my analysis, similarly to the t-test. I press next. I can now decide whether I want to use a critical p-value or use FDR. Let's remain here. I create hierarchical trees. Again, only the gene tree and save. Okay. And here we go with the results. And this Wilcoxon-Mann-Whitney test has found much more. Has found 489 significant genes, and we can review this also here. We have to concentrate again on those groups that we have chosen for the comparison. Treatment 1, 30 minutes, which are here in this area. Treatment 1, 60 minutes are here, and they have changed in a number of cases in accordance to this test. So this is 2 different approaches to find interesting expression profiles. One is the t-test, one is the non-parametric test, Wilcoxon-Mann-Whitney. Let me continue with the presentation. DECODON also provides the possibility to combine the results from different tests, regardless where they come from. This can be different flavors of the t-test, different non-parametric tests, it doesn't matter. You can store your results as clusters and then run operations on these clusters. You can, for example, use the union, the exclusive or the intersection operation to either combine with the union the findings, find out which finding is due to exclusively one or the other test that you are studying, or you just find the intersections or findings that have been found in all the different tests. Then we have another scenario created to just show the usefulness of Pavlidis template matching, PTM. Pavlidis template matching makes sense, for example, if you have read a paper which describes a similar approach as you are interested in, and they found a certain protein with a certain dynamic behavior for the conditions that you also study. And then you want to find in your experiment those expression profiles that behave similar to the published protein. Or you have found one protein in your data and you want to find all those other expression profiles that belong similar as this certain protein. For this you can use Pavlidis template matching, and how this is done is what I can show now. I just click the PTM dialog And then we have access to a list of all of our expression profiles. If they are labeled with spot IDs, you see here the spot IDs as labels and the respective expression profile across all the 48 images. And then I could choose a certain one which has a typical behavior that I'm interested in. For example, let me go up. For example, this one, we see that there is a nice high expression level for, as I know already, for the cell lines control and treatment 1, then a very low expression level for treatment 2, and then a high one again for treatment 3, across all the points in time. And we can use this one to use this as a template and find other expression profiles that match this behavior. I also want to construct a hierarchical tree. I have to enter a p-value. Let me just use this 001. Maybe we will find some. If it's too restrictive, you can redo this analysis using a different p-value. And then I press OK, change my clustering settings again, and then get a nice new result tree which includes similar expression profiles to the one that I have chosen as a template. You can see this also in the expression graphs. There is the mean expression graph shown with the symbols here, with the dots. There is the sample, the template, shown as the cyan line. And then you see in gray all the different expression profiles that match this with an error rate of 0.001. Similar to the t-test p-value. Thanks again for your attention. It has been a pleasure. Bye bye.